Welcome to the Neural Implant Podcast, where we talk with the people behind the current events and breakthroughs in brain implants in an understandable way, helping bring together various fields involved in neuroprosthetics. Here is your host, Laden Yurachek.
Hello, everyone, and welcome to the Neural Implant Podcast. Today's guest is Dr. Kevin Otto, and he is an associate professor in the field of neural engineering, device tissue interfaces, and neurostimulation. So we talk a lot about his research with implantable devices, and especially in the peripheral nervous system, and how these devices basically go to failure, and being able to avoid these devices going to failure. So he's also doing some brain circuit mapping, so we talk a little bit about this, as well as the goals of this. So hopefully you will enjoy this, and we'll talk more on the other side.
We started originally like a lot of people in the field, and in fact, I think the history of this podcast, I really was enamored by the brain and was fascinated by brain implants and wanted to design and use devices to understand learning and understand how the brain worked and help individuals who had disease or injury of the brain. What we've since realized is that brain implants are very difficult, and that the brain is a very difficult organ to understand, but at the same time it is connected to the rest of the body by nerves, by cranial nerves and spinal nerves, as well as and specifically within the cranial nerves by the vagus nerve. So the fun thing about being a scientist, engineer, and researcher is you have the ability to be flexible. So we have implanted a lot of devices into the brain, and as we started to use them, we got frustrated by our ability to use them reliably for long term to accomplish the tasks we wanted to do. So we started to investigate a lot of the body's rejection, how the device fails, how it doesn't, how it breaks. And so when we started looking at peripheral nervous system implants, what we realized is that many of the same problems exist. We want to be at the single cell or even subcellular level to interface with these tissues. And to do that, we have to penetrate and get next to the devices or next to the cells. And so that results in a rejection by the body, as well as more problems with devices lasting a long time. So we've been looking at how to make the devices last longer in the peripheral nervous system as well, which includes the vagus nerve, as well as other cranial nerves, somatomotor nerves, spinal nerves.
Is it more difficult in the peripheral nervous system to is there more rejection or less rejection in the peripheral nervous system?
There are advantages and disadvantages to the periphery. From one perspective, there's been a long history of interfacing with the peripheral nervous system, partly due to its accessibility. Generally, it's easier to find peripheral nerves. However, there's been a lot more focus in the central nervous system, maybe because of the disease and injury states there. So one of the things that's really important is the body of work. And so maybe the body of work isn't as quite as big in the periphery to build upon. So one of the challenges in the peripheral system is to understand what happens. What are the cells involved? What are the molecules involved? What are the approaches to the nerves? A major challenge is that oftentimes we don't even know where these nerves go when they go to an organ. We know that they enter the organ, but we don't know what they do yet. In addition, that said, there's a lot of accessibility. It's easier to oftentimes to get to periphery, which greatly facilitates the research, because then maybe the barriers to human level research are not as high. Oftentimes you could do a percutaneous or a simple skin incision where someone may not be as ready to have a brain surgery
Yeah, definitely. That's a quite risky thing. Like, hey, I'm just gonna open up your skull and take a look. I'm gonna put something in. Hopefully everything will be good. Oh man, but yeah, for the body, for the peripheral nervous system, that's not as big of an ask. It's like, oh, let me cut open your arm, cut open your neck, and put something in. That's a little bit less of a big ask, I guess. Yeah. So, what are some of these? What are some of these methods that you guys have developed, and what are some of the tricks?
Some of the tricks. Well, one of the methods I think that we really try to focus on is trying to understand the device tissue interface as an entire unit. That it is both sides of the interface, and we know that devices fail. Some people call that the abiotic failure modes. But we also know that there is the body reacting to the device that can result in failure of communication, whether that is from cells you want to talk to, either moving away or being pushed away or dying due to injury, or it's more of an encapsulation and scar tissue that builds up around the devices. So our approaches have been to really try to focus on the device tissue interface as a whole. And that involves looking at electrical aspects, looking at mechanical aspects, looking at chemical aspects, trying to keep the interface intact while we investigate all of these aspects, which really relies on a lot of novel and new techniques that are out there to traditionally, we're a very reductionist society. As scientists, we try to cut things very thin to look at small parts at a time. So it sometimes is very difficult to try to keep the entire system together as with as little manipulation as possible and still be able to investigate small parts.
Okay, so you're saying that maybe most scientists aren't looking holistically enough, and that they're looking too atomistically, something like this?
It's not necessarily a failure of approach per se, because what we try to build upon are techniques that have enabled the ability to leave entire systems in place and to do system level assessment, but then try to get down to not atomistic level understanding, if that's possible. So it's enabled by technology that didn't exist when many of these approaches were started. And within the technology and approaches, then you need things like big data and being able to take in a lot of data to understand how you might have, if many things are changing at one time, sometimes that can be really difficult to understand.
So do you think this will just naturally be solved later on by bigger computers and artificial intelligence and this kind of stuff?
I definitely think that there's a role for big data and machine learning in understanding multidimensional complex systems. That is something I think the next generation of scientists will automatically think of. Nobody has a paper map anymore because we all have Google Maps, and you can just use crowdsourced information from a lot of different sources that run through magical Google algorithms, and we know where to go, and we know exactly how long it's going to take to get there, as opposed to trying to do an estimate on paper of distance versus speed versus time of day and maybe how bad traffic could be. So the embrace of big data techniques is very advantageous for the field.
So the big theme of this podcast is glial scarring, and I'm wondering, how is it different in the peripheral nervous system versus in the brain?
That's a great question. We've done a lot of work in the brain and understanding some of the cells and the time course and the molecules, some of the fundamental engineering aspects that affect the degree of rejection by the brain. And what we've been finding is that actually a lot of similarities exist with the periphery. There are quite a few parallels with what the peripheral system, how it responds. Of course, there are different cell types and there are different molecules that are involved. So it's not clear yet exactly how different or similar the peripheral nervous system is. What we're finding more and more is that the similarities outweigh the differences, which is good because that gives us an idea that a lot of the approaches that we've been trying to help in the central nervous system are likely valid in the peripheral nervous system as well. So if we use smaller devices that are minimally disruptive, that are mechanically and electrically and chemically matched and active to help the peripheral nervous system both ignore the device as well as embrace the device, those techniques hopefully will help the reliability of interfaces in both cases, and specifically in the peripheral system.
Yeah, you're talking about the mechanical matching, basically matching the stiffness of both of the material of the body and of the device. Do you find that, and obviously that has to do a lot with movement, do you find that a lot of the peripheral nerves move more than the brain? I mean, I don't know, I kind of think of like your neck and your arms and stuff like this wobbling around versus your brain not so much.
Right. It is absolutely true. In many cases, the peripheral nerves move quite a bit more. At least we think they do, although there's a lot of evidence that certain brain structures move around a lot. Sometimes maybe during even shaking your head or maybe a subconcussive event, maybe you actually get a lot of movement of relative brain structures. The mechanical mismatch question / movement is very complicated because the mechanics of the situation are a factor of many different things. There is tissue movement, and the tissue is not homogeneous. There are local areas that are more stiff than others. But then on top of that, the size of the device matters a lot. If, in fact, a device is stiff but small, the mechanics are totally different than if it's larger and softer. And so arguably, it's difficult right now to say how the perfectly mechanically designed device would be. We have ideas and we have data. People have published data that smaller is better. There's some indication that softer is better. But what combination of size and stiffness is going to be optimal it's hard to say what the perfect design is, and it's probably dependent on where you're going to put the device, and it may be dependent on how old the person is or how many injuries they've had before. There could be a lot of factors involved in that.
Yeah, so for a very old person, maybe put one in the shape of a prune or something like this. I thought that was pretty self-explanatory. Like softer and smaller is kind of the answer. But are you saying that people are disputing which one is more important, like soft or small? But obviously, like soft and small would be the ideal, wouldn't it? And like as soft as possible. Like if we could get it to like snot level, then that would be ideal, wouldn't it?
Again, I think the challenge really comes as humans. I don't know that we are very good at changing orders of magnitude of scale or stiffness. Sometimes we don't think very well at that orders of magnitude level. So if you have something that is incredibly stiff, but it is incredibly small, it's not always clear biologically what that means. So I don't know that it's easy. I don't know that it's that definitive to say tiny little snot bubbles are perfect. I think that it's a very complex problem, and and one that may not be linearly dependent. They may actually have a high interaction term that may be nonlinear, and we don't necessarily know how the cells are going to respond.
Interesting. Well, as much as I would love to put snot bubbles into my body.
Well, and that said too, we're beholden to manufacturing in all of that too. That we can, on paper, design the absolute best device, but if we can't make it or put it in, or a clinician won't use it, then, or they can, maybe it's too hard to handle, and if it requires a multiphoton microscopy approach to implant the thing, then that's probably not even clinically viable. So
yeah, that was actually kind of my idea. I've been sitting on this idea of like hydrogels and 2-photon microscopy, not microscopy, but lithography of some hydrogels to basically make some structures, and they can have like channels inside, like fluidic channels for medication or whatever, and then it would be soft enough that you could have potentially an ion channel or something. I don't know. I was going crazy with this, but yeah, I thought that would be like the ideal. That would be the perfect version of an implant. What do you think?
Yeah, I think if we, one of the things that I like to emphasize in my research group is that we need to have, we're in biomedical engineering, and bio being the important thing, we need to have biologically inspired approaches. And if you think about the biology cares about things at the ionic and molecular scale. So we should be thinking at that scale when we're anticipating how devices in the body might interact. And if that is the design is at the molecular scale, then so be it. That's what we need to do, is think about how to best design devices that the body will appreciate at that scale. And it's difficult to manufacture. That's really, where you're talking about with maybe innovative and advanced manufacturing techniques that would give us molecular precision.
Well, okay, so I didn't mean this to turn into a verification of my pet theories, but what if you were able to have like a substrate of like polyimide, something that's a little bit stiffer, or even silicon, who knows, and put these snot bubbles on top of it. That way you have like a delivery system, but the snot bubbles kind of like buffer any movement. So the way I kind of thought of it was like a car seat. It's made of metal, right? But it has foam, so that you don't die when you hit a bumpy road or something like this. So you don't like have your clothes like wear off or something like this. What do you think about something like this?
I think that's so. I'll say that's basically the approach that we take in the lab. Is the first approach is to understand the biology and understand how the biology interacts with devices, and then once we have that knowledge, we can use that to make informed engineering approaches. And so major areas of research in the lab are looking at coatings of devices that may help the device tissue interaction, maybe keep the body from ejecting the device. Or I think one thing that's very important in there is that the car seat, the cushion, can't significantly increase the size or stiffness of the total device too much. So trying to keep your surface modifications small, but add the functionality that you're talking about, where you can cushion it mechanically or interface chemically, enable better electrical interfaces if that's going to be your mode of communication. I think that's, like I said, that's exactly one of the areas that we're looking at, is surface modification, device modification, so that we can cushion the device tissue interface in our way. One of the strongest effects of the characteristics of the device is the overall size of the device. And this is a very strong effect that smaller is better. And we work very hard to make the devices very small. And then if we modify the devices with coatings or with some sort of cushioning, and it makes them 3 times bigger, then we've, maybe not helped. So trying to make the devices using thin film techniques, using approaches that don't change the overall scale of the device is very important. But I guess, as you said, still trying to make the coatings bioactive, and maybe the next generation is to do things along the line of what you were mentioning, where the device is a part of this feedback homeostatic system. If it needs to grow and reach out to the cell that it needs to reach out to, then it can do that. It could extend chemical or electrical communication channels. If that requires mechanical capability, then it would, I don't know what if you think about a biological system in a homeostatic way, there are many, many little feedback systems that are at work, and we don't yet have those systems built into the interface of the device. On-demand drug release, on-demand mechanical changes, those aren't built into the interface. So incorporating sensing and actuating capacities onto the surface of the device, I think in the future will help to drive smart devices.
Interesting. Whoa. Okay, so on-demand drug release, that's pretty self-explanatory why that would be necessary. But on-demand mechanical changes, so like stiffness changes, how would that be? Besides like implantation or and then being in for a longer time. But are you saying like over like day or night, change or something, or how would that work?
Well, so I think previously you've talked with Walt Voigt, and they have mechanically adaptive devices, and there are other examples out there of mechanical adaptive that are typically to accomplish the immediate insertion this needs. They're stiff enough to be inserted, and then they soften in the body. Or you have some mechanical adaptation ability within the backbone of the device. We haven't yet incorporated how that might be used to continue to keep the device going long term, for example. So you could have microdomain mechanical changes that might make things better for the device tissue interface. And the corresponding thing can easily happen in the electrical or chemical domain as well.
Yeah, I seem to remember some kind of voltage regulated material that was able to stiffen or soften based on the voltage, and I don't know how fine they could make it, but yeah, I wonder if that would work as well.
Yeah, exactly. So Jeff Capadona and Dustin Tyler's work at Case Western, they did work on this. They were both people I was going to suggest you could talk to. I know you've talked to a lot of Case Western people already, but they had voltage regulated mechanical backbones for devices.
So you also work on mapping brain circuits and where the nerves end up inside the organs, right? As we were talking about earlier.
Correct. I think what we're really driven to the goals of the research as a whole. I didn't start out in the field to understand the device tissue interface. We were driven here because the interface was limiting our ability to ultimately help people and understand the brain and neuroscience. So our progress in the lab typically is to have parallel projects of prosthetic system utilization design, use what we have to do what we can, as well as at the same time in parallel try to make the interfaces and make the devices better. So it's a bit of a design loop where you make it better, go back and test it, make it better, go back and test it. So within the testing aspect right now, we're working on a DARPA program to understand if we stimulate the peripheral nervous system to try to improve cognitive performance, and we do see cognitive performance improvements, where in the brain is that being observed? What are the the elements of the brain, the nuclei, the cells that are changing to enable that improvement in performance.
Interesting. And so how are you going about finding this?
So in these experiments, we have ongoing animal experiments that are using behavioral measures of cognitive abilities. And in those cases, then, what we're going to use are peripheral nervous implants. The initial studies are being done on the vagus nerve, which at this point we're experimenting. The parameters of experimentation are timing of the stimulation and aspects of the electrical current relative to the behavior. And once the animals have shown that their cognitive abilities improve, we'll use early gene expression to look inside of the brain to see where the plasticity in the brain occurred.
What's the resolution of that?
Our approaches right now are both to use light sheet microscopy as well as multiphoton microscopy. So in both cases, the light sheet microscopy allows much larger sample to be imaged all at one time. Multiphoton, we can get higher resolution, but you have to use a little bit more confined sampling. But in both cases, we're at the cellular level with multiphoton subcellular. Our initial observations with the light sheet are to look at specific nuclei, brain regions that are affected, and once we know where to go look, then we can look at multiphoton to understand if there's subregion, subcells, subcellular populations that are affected.
I mean, it's a little bit obvious, but what are your big goals with this research? What do you hope to bring to the community?
Right. So yeah, we're very grateful to DARPA. This is a DARPA-funded program, and was originally administered and initiated by Doug Weber, one of your past interviewees. So our goals are to really facilitate the DARPA goals, which are to understand what is the role of the vagus activation in the drive of the brain and learning and cognitive abilities. It's always a fascination of mine to understand how do we learn and how can we learn things faster, and how do different people learn things, how even does an individual, I know that I've had times where I felt like I learned things very fast, and then other times where I wasn't able to focus and wasn't able to absorb. And why is that? So I think sort of a fascinating component of this research is to understand how does the autonomic nervous system really engage your brain to really tell your brain, this right here is really important. You need to pay attention right now.
Yeah, definitely. That would be a godsend for most students.
It's a technique that we use in the classroom all the time, is try to tell the students what I'm about to say is extremely important. And maybe this is a way to engage those systems electrically to try to communicate with the brain on a scale the brain really appreciates.
Yeah. Yeah, that's better than bringing poisonous snakes, I guess.
That might work.
Interesting. And so do you think this is the best way to go about this? Like finding how best to learn? I mean, it does seem pretty interesting. Focus and everything like this in terms of learning, but I also found like being able to relate some of the information is very helpful. So for example, I love languages, and when I'm learning a Turkish word, it's like I have no idea what it is. But if I'm learning French word or something, I can put it into context a little bit better because it's more similar to what I've seen. Are you able to measure something like this?
I think that's a fascinating question. I'm lucky enough to have an outstanding team to work with from here at the University of Florida with the Department of Neuroscience and with some of the VA investigators. I think the levels of cognition that you're talking about, what is the level of learning something in terms of associative versus even higher levels, are well outside of my expertise, but my team has the expertise to do this, and they've designed behavioral approaches to evaluate levels of learning, levels of cognition that maybe this will only work for certain aspects, though that's not what we think. So it's a fantastic question of at what level can you teach the brain to do something? Can we actually get around some of the initial just associative and aspect? Right?
And so this is kind of a controversial question, I guess, is what do you think like a big mistake or wrong direction that some researchers may be going down?
That is a controversial question. Fundamentally, I don't necessarily think there are mistakes or wrong directions in research per se. It's research for a reason. If we knew the answer, we wouldn't do it. It wouldn't be called research. I think so. In a perspective sense, misdirection is difficult to assess in research. That said, I think it's the best directions are enabled by knowing the history and the literature, and which you can get in the information age really easily, but then also communicating with your fellow researchers as much as possible. So trying to understand what's been done and what's failed. 30 years ago, you just had to be around for a long time so that you knew everybody and you knew everything that hadn't worked. But now it's a lot easier to know the history, and there's even scientific blogs about things that can't be published because they're negative results, but everybody knows that happened and that maybe it didn't work the best way. So
interesting. So get more stuff out in the open, something like this.
We as part of a reading course that I instruct, we always do a sort of a VH1 behind the science aspect to it. So if there was a last page where the authors could tell you what really happened, what could they put in the paper that isn't in the paper? And so we always try to fill in those gaps as much as possible. Try to understand who are the people that did this? Where are they? Where did they come from? What do you think they tried before the final data that ended up in the paper? And how did the paper get to the polished form that you read it when you're reading the journal? And sometimes that's a really fun part, is then you can go to a meeting and you see that person and you actually ask them, what really happened? Can you elaborate on their paper?
Yeah. Interesting. Yeah, that's kind of the purpose of this podcast, is like a less formal, transmitting some less formal medium of information than a paper. So if you had to say, if you had to talk about your behind the scenes, what would you say about it? I mean, you can maybe omit the cocaine binges and I don't know, like the band falling apart, or I don't know, whatever, but whatever's typical for the typical VH1 ones. But what would you want others to include, and could you talk about that?
Wow. Yeah, that I didn't anticipate you flipping that around.
It's like, we want more of this. Now I have to do it. I don't know.
So I do think one of the things that I emphasize in my group a lot is to really understand the personal drive behind science. Why do people use a certain technique or a certain implant? What's their history? Why do they use a certain recording system or a certain stimulation system? Is it a lab that they came up in? Is it a location of where they live? Is it the continent that they're doing science on? Or do they have industrial pressures? So, I don't think there's too much hidden about our science. We use a lot of techniques because of where we are, where we came from. I don't think there's a lot to tell people outside of what's in the papers that I can think of.
Okay. That's fine. You don't have to say anything. Yeah, it was really interesting. So I've done an interview with Harbi Sohal, and as of this moment, it hasn't been published yet. But by the time listeners will hear it, it will have already been published. But yeah, he was saying that he had a big motivation because he had both of his parents be very affected by some of this stuff, and you know, in the future, very short future, it could be you know, cured. It could be as simple as, you know, the antibiotic or something like this, and basically having this inflammatory response reduced, you know, in bioelectronic medicine. So is that something you're talking about, or were you thinking about something else?
Yeah, that does indicate one area we haven't talked about yet. About 3 years ago, I was actually diagnosed with adult onset type 1 diabetes, which is sort of a very interesting disease for a biomedical engineer to get, because there's all kinds of fun devices and experiments you can do on yourself now. But it definitely, it couldn't have corresponded better in terms of the field, in terms of bioelectronic medicine and the drive to understand these indications. So we have a major research effort in the laboratory looking at the use of bioelectronic medicine in diabetes. And it changes your personal approach when the impact is closer to home, at least potential impact.
Yeah, definitely. So I guess there's a silver lining to this, you know, fewer gummy bears, but more gadgets and toys to play with.
Yeah, gummy bears are right out. But you're right, the insurance company does pay for gadgets and toys.
So many people are just starting out in this field, and we talked about this a little bit before we started recording. But what recommendations do you have for people who are just starting out in this field, and how would you kind of suggest that they go about this?
Yeah. So, you know, first off, obviously, people are listening to this already, so I couldn't advise anyone to go in any other field. It's a fantastic field. And one thing I guess I would start with is, what do I look for in my lab members and or collaborators? And, you know, the most important thing is really enthusiasm, being excited about the work and enthusiastic. And the fact that people are listening already means that they're engaged. And then creativity. You can learn a lot of book smarts, but being creative and trying to think outside of the box just for fun. But so in terms of what to do, we knew this question was coming as a lab, so I asked them, you know, what do you think? And by far, all of them said read, read. So you know, I think it's important to try to read what you can. Read papers. If you're new to the field, the papers are difficult, but websites that are a little more informal, lab websites, are extremely useful. They try to break it down a little bit more. I also think it's important to read the biographical status of people. Where did they come from? If you want to be a grad student, you should learn about some grad students. Some other things that I think are fantastic ideas, and they fit within this podcast really well, are to be a citizen scientist. Computational approaches, being able to write programs that might do things that you want to do someday. And then I know you talked with the Backyard Brain guys that, you know, a $99 spiker box, you can buy one of those and start doing your own neural implants. And then this is something that a lot of people might not appreciate, but volunteering in a lab is actually something really easy to do. If you have already that enthusiasm and that creativity, and you have a pretty good working knowledge, and you've kind of hacked at home, just sending an email to a local lab, even as a high school student or even younger. We've had younger students that we've been brought into the laboratory to show them what science is really all about. And all of those, that's a really nice pathway to start understanding what happens in a research laboratory and how it can turn into your life and career someday, if that's what you want.
Yeah, I think that's really accurate. I think the volunteering and just getting in there, because you can maybe tell me I mean, obviously if somebody's volunteer, how they work, how their disposition is and character and everything like this. But otherwise, the other option would be basically to send a blind resume, a blind email, right? And do you get lots of emails, and how many do you get?
A lot. I think most lab, the senior lab people are going to get a lot of emails, a lot of contacts, and it's important to. I advise people two things: be very formal and professional in that email contact. Include a resume. Include reason. Include those drive for why do you want to do this? What makes you unique, and why are you excited? And then the second thing is to be persistent. Email is not always answered, and everyone is pretty busy, so it's not. It's not uncommon for me to receive 6 to 10 emails from an individual before I finally have a chance to say, okay, this person really cares. I should respond.
Wow, that's crazy.
I'm not saying that's the norm, but if I see something like that where somebody is really driven, that I usually say we should talk.
Interesting. Yeah, that's I guess it's a good filter as well. Like, is this just a blast? Is he just blasting out emails to everybody, or is he really interested in this lab? And yeah, that's really interesting.
Well, and the other thing too, I'd say is absolutely do not discount the lab members. Contacting the lab members directly and saying, hey, if you're around, just say, hey, do you mind talking with me on the phone? Or if you're around, say, could I buy you a cup of coffee? I just want to know what it's like. They can give you a lot more interesting information usually.
The dirt.
Exactly.
Cool. Well, Dr. Kevin Otto, this has been a pleasure. Thank you so much. If people want to find out more about you, how do they do that? Or is there anything else that you wanted to cover that we didn't cover?
No, I think this was excellent. Thank you very much. My email is pretty easy to find, and in particular here at the University of Florida, our lab website is nprlab.org. Much of the information we talked about today, as well as a lot more, all of our papers and my contact information is there.
Nice. Just remember to email 10 times.
A LinkedIn contact is also extremely useful. So,
okay. Thank you so much. This has been a pleasure.
Thank you.
Hey guys, hopefully you enjoyed that. Yeah, it was really interesting. I mean, I didn't mean for it to turn into like a verification of my pet theories, but it did a little bit. And I wish there was more specifics of what exactly was being done. I feel like he kind of danced around the topic a little bit but at the same time, I think it was very helpful. And at the end, man, I always feel like I feel like I'm the last person to find out about stuff. So he told me about a really, really cool website called neurotree.org. And this basically shows the collaborations or the associations from all the people. So you could see who came from what lab and then what lab they created and blah, blah, blah. So it's like a family tree, but for scientists. And I'm like, whoa, this is so cool. So I don't really know if it affects you guys that much, but it's great for me because it's like a treasure map of potential guests. And I'm like, ooh, this person, this person, this person. And I see how they're related. And it's really interesting. But yeah, maybe it could be interesting for you guys as well. I think there is a wiki as well. And this is something that I've been thinking about doing, is maybe putting together some kind of wiki for neuroscience and neurotechnology, neuroimplants, and being able to basically collaborate. Everybody put together their information, and maybe we can bypass the journals. Maybe we can still use the journals for formal publications, but for the basics and everything like this, we could have a wiki, maybe YouTube page. I'm thinking about putting together a YouTube page eventually, putting together interviews and everything. And yeah, we'll see how it goes. I'm really interested. I want to learn and I want to teach at the same time. And yeah, so hopefully you guys enjoy this. Hopefully you guys enjoy what I'm trying to do. I know that Dr. Kevin Otto was a big fan of the show, and it makes me feel very nice. It makes me feel very good that I have such a big fan and that turns into a guest. And yeah, I really hope that you guys are listening and you guys are fans as well, and that maybe you'll be on my show one day. Doesn't matter if you're a professor, doesn't matter if you're a PhD student, or even like a high school student. You know, if you're 15 years old, maybe you can be on my show one day, and you'll be like, oh yeah, this guy invented laser flamethrowers. It was amazing. So until next time, think about this idea, laser flamethrowers, and what it would mean for society, and we'll be back next week.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.